The overall goal of this procedure is to follow the dynamics of apoptotic cell clearance in the Drosophila embryo. This is accomplished with transgenic flies containing a simucytoplasmic GFP marker, which labels phagocytic cell populations. Embryos with the simu-GFP marker are collected, washed, and dechorionated. Next, the embryos are positioned for injection, followed by monitoring of the CNS. The third step is the labeling of apoptotic cells by injecting the embryos with specific apoptosis phagocytosis markers, such as an exon 5. The final step is to make time-lapse recordings of the embryonic CNS using confocal microscopy. Ultimately, the results can show the localization and behavior of apoptotic cells and phagocytes during different stages of apoptotic cell clearance through fluorescence confocal microscopy.
The main advantage of this technique over existing methods like immunohistochemistry on fixed embryos is that we can follow dynamics of apoptotic cell clearance.
Apoptotic cell clearance is a highly dynamic process containing diverse steps. This technique allows us to follow the different steps using specific markers for apoptotic cells and phagocytes.
The protocol begins with making a glue from conventional reagents. Unroll double-sided tape. Then, roll it back up to fit into a scintillation vial. Fill the vial with heptane and seal it with parafilm. Then, attach the vial to a nutator and gently rub.